So hello and welcome everyone. Good morning, good afternoon to everyone, good evening to wherever you're joining us from. My name is Evangeline Arithwala and I'm the Alumni Engagement Officer. Today I also have my colleague Jin Ganchu who has joined us today and will be managing the waiting room. To introduce our speaker today, if we can please go to the next slide. Thank you. To introduce our speaker today, we have with us Commonwealth scholar Bovenil Anye Cho from Cameroon. Bovenil will be presenting this webinar titled Synergizing Biomass Growth Kinetics and Transport Mechanisms to Simulate Light or Dark Cycle Effects on Photo Production Systems. As mentioned, this webinar is part of the Climate Action webinar series. To give a brief introduction about our speaker today, Bovenil is currently pursuing his PhD in chemical engineering. And prior to this, he was a research assistant at the Stellenbosch University in South Africa. This is where he also graduated with a master's in engineering from the Department of Process Engineering. He's passionate about entrepreneurship and research. I'm also pleased to inform you that Bovenil is the winner of this year's Research Impact Award Topical Prize for Climate Change Related Research. And today in this webinar, he will be presenting on his winning research publication. Bovenil is a 2019 Commonwealth Scholar from Cameroon. He is currently pursuing his PhD in chemical engineering at the University of Manchester. So without any further ado, I now invite Bovenil to begin with his presentation, and on behalf of CSE, Bovenil, a huge welcome to you. Over to you.
Thank you very much, Eva. Hello, everyone. Greetings wherever you are in the globe. As Eva did the beautiful introduction, I will just move into what we're about to discuss. But before I go any further, I would like to just mention, as the topic reads, it's a little bit technical, but I've tried to put in some pictures and some animations to make the content delivery user-friendly for people who are not into engineering or physical sciences. So without wasting any more time, let me introduce the outline of the presentation. So I have structured the presentation into 6 main sections, starting with the motivation, moving into the results, the methodology used, presenting the results obtained, discussing the future work and just showing applicability of the work, also presenting my CSE scholar experience, and finally providing some relevant references. So with that said, let's move into the first part of the presentation, which is the motivation. This picture speaks volumes about why I'm here today and what I'm doing. This is mainly because the picture is taken from my hometown, which is Buea, southwest region of the Republic of Cameroon. So I am among the people whom you see here. And from the picture, I can draw out 3 characteristics features. The first one is solid and liquid waste generation. The second one is rapid population growth. And the third one is infrastructural development. Now, these 3 characteristics are not only common to my home country, Cameroon, but they cut across several, if not all, developing countries, whereby waste is an inevitable consequence of rapid industrialization and urbanization. As we all know, improper waste management often results to serious public and environmental risks, such as the prevalences of diseases such as typhoid, malaria, etc., etc. Also, the continuous dependence on fossil fuel-based energy sources to meet up with the energy demands of these rapid developing countries has brought about serious global environmental challenges, such as resource depletion, environmental degradation, and energy shortages. So there is a need, or let me specify, there's an urgent need for clean and renewable energy to meet up with these energy demands such that we can have electricity generation, transportation, and heating in a sustainable manner. With this in mind, my target or our target is to achieve this clean and renewable energy by simultaneously treating waste organics while producing clean water and energy. Now, to make this concept visualize, I also put forward this sketch here, this schematic where you can see a typical source of waste stream, in this case, industrial wastewater, being biologically treated by two major techniques, dark fermentation and photofermentation. Now, the main difference between the two, or let me rather start by saying fermentation is just a process whereby a microorganism can, metabolize or can feed on the organic portion of the waste stream. And in the process, they release byproducts. Some of these byproducts are biohydrogen, which we are interested in, and a little bit or a little amount of CO2. So because the process can require light, as well as it cannot require light, it's often termed dark fermentation when the microorganism doesn't need light, and photofermentation when the microorganism requires light. Now, having understood like there are two biological approaches to handle this waste and produce our energy, which is our biohydrogen, and the residue is just, the water itself that you can use for agricultural purposes or even any other reusable manner. So having understood this concept, I also want to point the fact that what you see here is an indication of a container. Let me put it, a container which houses the mixture, the waste, the microorganism, everything in there. So we call that container a bioreactor. And for dark fermentation, it's a simple bioreactor. But for photofermentation, it's called a photobioreactor because it has an additional requirement of light. So we are going to focus this presentation about these, containers, because we are trying to see how we can scale up the process and like increase the amount of waste that we are treating, as well as the amount of biohydrogen that we are producing. So, but first, it would be nice for us to see how a wastewater treatment plant looks like. So typically, this is how it looks like. And when I say a wastewater treatment plant, I would like not to limit your thoughts. Also think about sewage. Sewage sludge is also treated in the same manner. So the technology that we propose can handle all of this. So it's going to look like this. And by the end of it, we'll be achieving 3 of the UN Sustainable Development Goals. That is Goal 6, which is clean water and sanitation, Goal 7, which is affordable and clean energy, and Goal 13, which is climate action. So I guess by now, you're beginning to see the significance of the work, why it was awarded the 2021 CSC Research Impact Award for climate change related research. So, but first, let's go now into a little bit into the technical aspects of the work. So if we revisit the two methods of handling this waste, there's the dark fermentation and the photofermentation, and we do a comparison, we realize like the photofermentation approach is very efficient with almost 90% conversion rate. So one is tilted to go towards the photofermentation in such a way that residue, or let's say end product from the dark fermentation, is further photofermented so that you can get like maximum biohydrogen from the process. So now the obligate requirement of light by this photofermentation process poses significant challenges. The first one is light attenuation. So this concept of light attenuation is something that you're going to be hearing throughout this webinar. But for now, I would like to just put it into perspective so you can see it. So light attenuation is first of all defined as a physical phenomena. Let me use a physical because you can actually see it. So the incident light intensity decreases within the reactor. And what do we mean by that? So at the start of the process, we have our Remember the container I said, right? So it's called photobioreactor now because it can allow light to come through. So our light rays are coming through. And we have in there our waste organics, as well as we have our microorganisms in there. At the start of the process, time t equals to 0, everything is fine. There's even distribution of light within the container, within the reactor. But as the microorganisms are happy, they grow. So the culture becomes dense. A few days later, just let's say about 4 or 5 days, the culture becomes heavily dense. And that physical phenomenon, you remember about light attenuation, can be seen here in such a way that the incident light intensity, you see this portion is having light and this portion is dark. So 90% of the light is absorbed within a few centimeters of the reactor and the rest is remain dark. And this has consequential effects on the overall productivity of the system because the microorganisms require light. So if they're in the dark region, they are not getting light, so they are not growing. Instead, they are using some accumulated carbon source or some storage of energy to maintain themselves to stay alive, which we don't desire. We actually want them to convert the waste into our biohydrogen. At the same time, cells which are within this region of light, the light intensity is so high, so it's so harsh, so they will experience a kind of phenomenon we call photoinhibition. So the light inhibits their growth. So it doesn't kill them, but it inhibits them. It makes them inactive, so they cannot produce our desired biohydrogen. So now, this phenomenon is not specific to this bioreactor configuration because you see this is a tubular photobioreactor and this is a flat plate photobioreactor. So the observation is common across all photobioreactor designs. And therefore, there's a challenge now to upscale this technology. Despite the high productivity rates we can get from it, we cannot scale up the process because usually we want to increase the diameter as well as increase the height. And that will mean we'll be wasting energy into the system since as the light will not be going to the end. So this constitutes what I'm going to be presenting today, and it's the focus of my PhD research. So having discussed this, let's go into the methodology section and see what I've been trying to do to address this issue. So, but first, to understand what is happening, we need to zoom down into the photobiotechnological operation and really see like what's happening. And within this slide, we can consider one photobioreactor design. This is a closed photobioreactor, which is a flat plate. And we see that the process itself, it's a multi-time and multi-scale phenomenon. By that, I mean, if we look at the light field, radiation, the time taken for the photosynthetic active units. So these are units, or these are pigments that can absorb light, and that light is used for growth. So the time taken for them to absorb that light occurs within milliseconds. But the time taken for that absorbed light to be used for cell growth occurs within a few hours to days in some microbial species. Now, that time difference from milliseconds to days creates a time scale, which is very different, which we need to capture. Also, within the cells, the metabolic reactions are occurring in a micro scale, while mixing of the reactor and fluid dynamics, everything is in a macro scale. So we have also a micro scale and a macro scale. Now, if we couple the multi-time and the multi-scale phenomena, the light attenuation becomes very challenging to deal with or to handle. Now, the major thing is that cells within certain positions in the reactor are going to be moved around by fluid dynamics. So the time spent within the dark zone and the time spent within the light zone are different. So the cell experiences some light dark circles. And the challenge is to see how to balance these circles, because if the cells spend so much time in the dark, they're not going to produce, they're not going to convert waste. If the cells spend so much time in the light, they're going to be photoinhibited. So we want to balance it in such a way that when the cells capture the light, they should be moved into the dark region so that they can use that light efficiently for photosynthesis. And then they are brought back to the light so that they can capture more light for photosynthesis, like just balancing up the process. Now, to be able to understand what is happening in this process, we need to look at literature, what has been done, where are the gaps, and what do we need to focus on. So when we look at literature, there has been several mathematical models trying to describe the relationship between photosynthesis and light intensity. And these models can be grouped into three main types. So type 1 models describe photosynthesis as a function of incident light intensity. So it means just light at the front of the reactor. They do not account for the light gradient, which is light within this part, as well as they do not account for the light dark circles, that's the cell's movement within the reactor. Therefore, these models are not desired for us because we want to scale up this operation. So we want to increase the diameter, we want to increase the height, we want to convert so many ways into our desired biohydrogen. Now, type 2 models take into account the light within the reactor, so the light gradient. But then they do not account for the light dark cycle. So the cells moving from dark to light and light to dark, they do not account for that. As a result, they can overpredict photoinhibition, which is the duration spent in the light zone. But 3 models can account for both the light gradient as well as the light dark cycles, the cells moving within the reactor. And they are the most accurate models and of interest to this work. And these models are based on the concept of photosynthetic physiological state models on the photosynthetic active units. So photosynthetic active units will be the units that can capture the light. So we can postulate models such as you can exist in separate states. In this case, 3 to 4 states, but in literature, you can have from 2 right up to 4. I've just illustrated a few here to make the whole concept to be visualized. So, but now, amongst these models in literature, there's one particular one which really stands out and it's very relevant to this work. So this is the 3-state, 3 means 1, 2, 3 photosynthetic active unit state model that was proposed by Eller and Peters in 1988. Now, the model assumes that the photosynthetic active unit of a cell, of a microbial cell, can be in an open state, closed state, and an inhibited state. So the probability for it to be in any of these states equals 1. Now, in the open state, it's inactive, but it can capture light and become active and move into the closed state. In the closed state, it has 2 options. Because it already has light in it, it can use that light for photoproduction, and then it returns back to the open state. Otherwise, it can capture more light. Since it was already light saturated, it captures more light, it becomes inhibited, and it moves into the inhibited state. Now, in the inhibited state, it can slowly recover back into the closed state. Now, if we can describe this state transition within the reactor, then we've solved the problem. We can describe how light intensity is affecting, the cells as they're growing, and we can see how we can solve the issue. So this state transition, or this transitioning among these states, is described by 3 ordinary differential equations. However, the challenge is that these reactions are happening within the cell, so they're in the micro state. So in the micro state, we also have macro state where we have mixing and free dynamics in the reactor. But how do we account for this within the macro state? That's where the challenge comes in. And in literature, there has been one study that stood out, which attempted and successfully coupled this together. In their approach, they assign convective and diffusive terms into these PSU states. So they can now simulate the state as the cell is moving within the reactor. In their approach, they were able to do this, and although it was computationally expensive because we're solving 2 biological transport models, and as well as different time scales, integration time scales, they achieve good results, satisfactory results. But we looked further into the study and discovered the coupling approach was not correct. it has like issues. Reason being that it defies the biological law because it considers that these states are not confined within the cell. So they are almost like outside of the cell and they are just moving randomly within the reactor. But according to the biological law, the states are confined within the cell and then the cell moves within each position in the reactor. So it's now clear, like it's now understood, like it's still unclear how hydrodynamics or fluid mixing in the reactor directly affects this photosynthetic active unit states within the cell walls, not outside of the cell, within the cell walls. And this led us to proposing a hypothesis. Before I go into discussing the hypothesis, it would be nice to explain what's happening. So if we consider the light field within the reactor, we discover that, or we observe physically from experiment that cells that are here and cells that are here are receiving different light intensity, or cells that are positioned anywhere within the reactor receive different light intensity at any point in time. But as the cell grows, so one cell at one position, next time it will be somewhere here, the other time it will be somewhere there. So throughout its history, it experienced a light intensity, which is different from the light intensity it receives when it's at any point. So based on this, we can say hydrodynamics, which is mixing in the reactor, can actually influence the cell as they are moving and directly affect the PSU states. With this hypothesis, we're able now to formulate 3 new ordinary differential equations, which captures the effect of fluid dynamics on the PSU state within the cell walls and not outside of the cell walls. So by considering pseudo-steady state and solving this equation, we end up with the macro scale growth kinetics. So macro scale now is now in the scale where we have now fluid dynamics and mixing and nutrients in the reactor and our waste organics. In this approach, we see like the model that we get has parameters, which are actually the PSU light and dark reaction coefficients. But when we compare this model with literature model, we discover like it's very similar. That's the substrate saturation coefficient and the substrate inhibition coefficient are actually ratios of these coefficients of the light-dark reactions. And therefore, only one parameter, which we call eta, which is eta, but we call the effective light coefficient, has not been defined here. So according to our approach now, we come up with one parameter which needs to be described. And this parameter is actually responsible for the cell light exposure history on the macro scale growth model. So at this point in time, one can think we have done it, we've sorted it out, everything is fine. But you remember that we are dealing at any point in the reactor, but we've not considered the effect of light attenuation, like the distance from the front to the back of the reactor. So in order to incorporate that, we need to incorporate a model that can describe how the light intensity decreases with distance in the reactor. So the famous model in literature is called the Beer-Lambert law, which says the incident light intensity exponentially decreases with distance with like the light path length of the reactor due to gas bubble scattering and cell absorption. So when we take this model and we substitute it into the place of the light intensity in the equation, we end up with the final macro scale growth model, which is this one that can capture the influences of cell growth in the macro scale as a result of light attenuation and light exposure history in the reactor. Now, the model looks fine and okay, but still we don't know how this light exposure history directly affects the macro scale growth kinetics of the model of the cell. So in order to do this, we design scenarios to investigate, studies to investigate. So the chosen photobioreactor is the flat plate photobioreactor because it has a high surface to area volume ratio, so it can easily be scaled up for industrial purposes. So when we consider the flat plate photobioreactor, we have 2 test scenarios. Scenario one, where we are controlling the influence of hydrodynamics in the system. So we have a spiger, which is just a device that can allow bubbles to go into the reactor. As the bubbles are rising, they are displacing water in their path and creating chaotic mixing in the system. And when we change the configuration from one spiger to two spiger, we can actually increase the influence of mixing in the system. So we are interested in seeing how this increase in mixing the system affects the cell growth in the macro scale. So we investigate different inlet gas flow rates from 0.1 to 2.0 meters per second, as well as different photobioreactor configuration, one spiger versus two spigers. So this is good in terms of the design of experiment. But now, how do we actually visualize the results? So we use computational fluid dynamics. And for computational fluid dynamics, we have to solve this partial differential equation. So this is actually the biological transport equation, which can say the cells in the reactor are moving as a result of some velocity factor pushing them around, as well as diffusion factor, which is spreading them around, and the growth or the destruction term in the reactor. So one thing to notice here is that the effective light coefficient is not included in this model because this model is solved at each point in time. But now we still need to be able to capture the effect of light intensity and light exposure after a certain period of time when the cells have grown. So we do parameter estimation, but this time the macro scale growth model is presented here with the influence of both cell distance, which is like the attenuation distance of the reactor from 0 to L, and also time. So we eliminate the distance component to simplify the model so that we can perform our parameter estimation. So we can estimate the value of this effective light coefficient in the system. But after simplifying the model is still highly nonlinear. So we use hybrid stochastic optimization algorithms to estimate the value of these parameters and to be able to describe the influence of light dark cycle on the growth of the cells. So with such a result now, in fact, in presenting the results, I'll first of all present some cases. So I'll split the results into 3 sections. So this is section 1. And before I go even any further, the results of section 1, I've presented just the first case because the 2 cases, this is scenario 1, 1 spiger, and if you remember scenario 2 with 2 spigers, actually have similar trends. So by having similar trends, I'll present just the results for 1 spiger, and then we can make the assumption of what is happening in the second spiger. So for the first spiger, we see that there's a dynamic motion from the bottom of the reactor to the top of the reactor in a random oscillating plumb. And what we can interpret is that plumb is displacing water on its path and is creating mixing in the system as the process is evolving. Also, we compare the kinetic turbulent energy in the system, and it correlates well with the region where there's chaotic mixing in the system. And these observations are common for both scenario 1 and scenario 2. Also, we see that by increasing the gas inflow rate from 0.1 right up to 2.0 meters per second, the liquid phase velocity as well as the turbulent kinetic energy in the system increases. And all of this is in agreement with literature finding. So far, we are confident about the results. Now, looking at the second part of the results, which is the effect of gas inflow rate on biomass growth rate. This figure shows how the biomass grows from mixing condition of 0.1 to 0.2 meters per second. So what we see here is that, first of all, the trend shows a sigmoid shape, which agrees with literature and confirms that the multi-scale coupling approach is accurate. Also, we see that as the gas inflow rate increases from 0.1 meters per second to 2.0 meters per second, the saturation cell growth rate increases from 4.2 to 5.0 grams per liter, with an increase of just about 17%. Now, in order to explain this amount of increase, we look into the velocity in the light transmission direction. So we see that there's about a 300% increase in the velocity along the light transmission direction. And we can attribute this increase in growth rate as a result of increase or better mixing in the culture, since the cells are frequently transited from the dark region into the light region and back and forth. So having explained that, we also see that although there's about a 300% increase in that, the biomass growth rate increased just by 17%, which is not a dramatic value. And this actually means that, or this implies, these results were expected because at 0.1 to 0.2, the system was already operating in a turbulent state. So in a turbulent state means like mass transfer is not a limitation. The only limitation is light. So every other thing kept constant. The only influencing factor is just light. So it didn't increase by a dramatic value. Now, if we look at the third part of the results, where we want to correlate the effective light coefficient with the mixing in the system or gas inflow rate. So this figure actually shows the results from the parameter estimation, because remember, we are estimating this value from the data. And we can see that the model fits the data really well. And this is a clear indication that our proposed model can actually capture the influence of light-dark cycle on the macro scale growth kinetics. From the estimated parameter value, we can also look at how it correlates with the gas inflow rate in the system. So what we see is that generally, the effective light coefficient for both cases was greater than 1. And greater than 1 means that cells in the dark region do actually absorb light. But how is that possible? It can only be possible if they are brought from the dark region into the light region. And these results cannot be explained by any other model in literature. So it actually shows why this model is outstanding, because it can capture that effect. And also we see that by increasing the gas inflow rate, you also increase the effective light coefficient. So you're increasing the rate at which you're moving the cells from the dark to the light region. And it's also increasing the rate at which biomass is growing in the system. So the correlation makes sense and agrees according to our expectation. But then how can we use this information? It constitutes the next part now. So in order to be able to use this information, we correlate directly the effective light coefficient to the increase in mixing in the system. So the first observation is that the effective light coefficient versus increase in mixing in the system follow a power series law expression for both cases. This is actually in agreement to other observations in literature where transport phenomena related models are actually power law series, power law expressions rather. And the correlation shows us also that if you look at the index, which is just, let's look at the powers, like these values, they are different for both cases, scenario 1 and scenario 2. And it shows to us like you cannot use this data for scenario 1 to apply for scenario 2, but the model structure is actually the same. So the only thing you need to do is to recalibrate the model parameters. So from this, we draw a conclusion that for this model to be used for different scales of photobioreactor or different configuration and designs, you have to recalibrate these parameters. As well, there's also, our investigation was limited to 2.0 meters per second, but we acknowledge the fact that by increasing the gas inflow rate beyond this, the model might take a different form. So this model is only valid for within this range. Outside of this range, you might need to formulate a different model. So this is part of the future work which we are going to investigate. But let's move into the future work and see how this model is actually creating an impact in the field. So before I go any further, I would like to call us back to remember, like at the beginning, I mentioned like when we are starting the process, the light intensity, it's fine within the system. So we're getting almost a uniform light intensity because the cell growth rate, it's actually the cells are sparsely dense, you know, and everything is fine. So they're happy. But then because they're happy, everything is fine, they start growing. When they start growing, the cell becomes dense, becomes really dense, and the light becomes attenuated. So we get just light from here to here, and all of this side is dark. But then now, this light attenuation here can be addressed in two manners in literature. The first one is to increase the incident light intensity. So when you increase the incident light intensity, the stronger radiation is going to penetrate deeper into the culture. So it's going to go deeper into the culture. Also, the second approach is to increase mixing in the system. When you increase mixing in the system, cells in the dark region are going to be moved into the light region, and cells in the light region are going to move in the dark region. But in literature, only the optimal control of the incident light intensity has been proposed. The extent to which you can increase the input gas inflow rate into the system to maintain an optimum light dark cycle is still not known in literature. And this is where our model comes in, because it can address that for the very first time. So in order to explain how this works, now let's consider the model we propose, which is this. And we can describe the cell growth as a function of light intensity and their light exposure values. So first, from this, we can actually calculate the optimal cell growth rate, which is this, just by taking the differential of this, differentiating this equation with respect to light intensity. We get this, and we can resubstitute this out, and then we get this. So this is our optimum effective light coefficient, which is represented as a function of, or just a square root of, the substrate saturation coefficient, substrate inhibition coefficient, and the optimal light within the system. But then, how is this usable? So if we come back to this plot and we see this is our system, right? So we have light attenuation within the system. But first, we need to know how much cells do we have in this system? How much biomass do we have in this system? So we calculate the cell growth with this model. When we know how much cells are in the system, we can actually calculate the average light intensity within the system. So we calculate the average light intensity within the system. From the average light intensity, we can substitute it in here to get the optimal effective light coefficient. The question now that we ask is this optimal effective light coefficient the optimum we got from the model, or is it suboptimal? If it is suboptimal, let's say at the beginning, let's say at the beginning it's somewhere here, then we know our cells are not experiencing the best condition. So we need to increase the flow rate. So when we increase the flow rate, we are pushing the optimal value to somewhere here where the cells can grow happily. So ideally, we can iterate this process over and over until we operate at the optimum effective light coefficient value, which is actually delivering the optimum or the optimized light dark cycle within the reactor. So therefore, light penetration distance is no longer a challenge, because we can have any light penetration distance, but we can only control mixing to make sure all the cells are getting equal light intensity within the system. So having presented this constitutes the next part of the work, because this is the theoretical application on how to actually use this in real life situations. So at the time this work was submitted for the CSC Research Impact Awards, I had done only this, but as of now, the work has been extended. So until now, we have published again another paper. But this time, if you remember, the model was specific to a photosynthetic microorganism, which is a microalgae and cyanobacteria. But also, we have photosynthetic bacteria, which have proven in literature to be effective in treating very recalcitrant wastewater streams. So if our model works, it should be applicable to all of these microorganisms. So this work focused on a different type of photosynthetic microorganism, which is photosynthetic bacteria. And what we're looking at is to be able to capture the effect of not just light and light attenuation and the light attenuation distance, but also to capture the effect of temperature. Temperature is another, it's another parameter which is very sensitive, because in real life, these cells can be operated, or the bioreactor will be operated in the outdoor condition. And in the outdoor condition, we know like the temperature fluctuates as well as the light intensity throughout the day. At noon, you get the highest or the peak, and somewhere at nightfall, everything goes down. So if our model can describe the influences of temperature, influences of light intensity, light attenuation, and the light exposure history, then we can model and we can predict what's going to happen in the outdoor conditions. As well, we also incorporated the aspect of upscaling from the lab scale into pilot scale of the operation. And as we can see, our model agreed really well with the experimental data, and this led into this publication. So further and future work that I'm working on is on designing an optimal photobioreactor that can take into account all of this knowledge so far and make sure it can produce hydrogen or continuously maintain the optimal hydrogen production for a long duration. So having presented this part of the work, I will just go in now into discussing my CSC Scholar experience. So the first thing I would like to say here is it has always been my dream to become a leading researcher in the field of bioenergy and sustainable energy production. But I will also underline that without the Commonwealth Scholarship, it wouldn't have been possible, because I'm currently, I can't afford the tuition fees at not just tuition and accommodation fees at the UK's leading institutions, such as the University of Manchester and Imperial College London. So since the start of my scholarship in 2019, I can say I have harnessed solid mathematical programming skills using softwares such as MATLAB and ANSYS Fluent. These softwares present engineering tools that can be used to address 21st century problems. However, they require licenses which are expensive to purchase. And as a student in the University of Manchester under the Commonwealth Scholarship, these licenses are made available to me for experimental hands-on learning. So I'm truly grateful for that, because without that, I wouldn't have had access to them. Also, I've been able to expand my network within the UK, that is Imperial College London, University of East Anglia. Outside of the UK, that is South Africa, Stellenbosch University, where I did my master's degree, and as well as China, that's a Xiamen University in China. So this collaboration network forms a vital part of my future career, because I plan to establish a wastewater treatment plant and a biorefinery in Cameroon. So I already have a solid team of experts that I'm working with as of now. And finally, I would like to say, with this experience, I have been able to develop capacity on writing high quality scientific papers. As of now, I have 5 under my belt, and I'm still to publish more. So this actually reflects the impact Commonwealth Scholarship has played in me in developing my career as a young and emerging researcher in the field of bioenergy and sustainable energy production. So I want to thank the Commonwealth Scholarship for this opportunity, and this concludes my presentation, as these are just some relevant references for those who are interested. And thank you.
Thank you so much, Bhuvanil, for your webinar presentation on your winning research publication. I'm sure you're super proud of yourself for winning this title of the Research Impact Awards, the Topical Prize, and also sharing your PhD research and where you are at with your research study and what is in future pipeline for you. I would now like to open the floor to Q&A session. I would invite attendees to send in your questions via the chat box. If any of our attendees are studying a research topic in a similar line of study, or you are already working in a similar field, or if we have any PhD scholars currently in our attendees, if you would like to connect with Bhuvanil to ask any questions, please feel free to send in your questions in the chat box. We do have a couple of questions that have come in advance to the webinar via the registration form. So I'll be more than happy to bring them up and address this to you, Bhuvanil. But yes, attendees, if you would like to ask any questions, please send them via the chat box. Thank you. So the first question is, apologies, I'm just bringing up the question. Sorry, the first question is, why is biohydrogen consortia not been set up in every developing country?
Thank you for whoever asked that question. It's interesting. It's actually interesting for every developing country. I will not limit my answer to developing country, but to globally, to say the technology, it's very, very complicated because you're dealing with systems almost like human beings. You know, cells and microorganisms are very, very tricky to deal with. They're just like us. So the knowledge in the field is still emerging and it's still developing. So at this point in time, most people rely on systems which they've already understood, you know, but we can say government legislations and international, you know, like conferences like the COP conferences on climate change are actually imposing, you know, legislations on government to try to refrain away from some of these systems and look into alternative forms like us. So I would say, as of now, what I have presented here is not the end solution. There's a lot of work to do. So the technology is not yet mature. And that's why, that's one of the reasons, I cannot say that's why, that's one of the reasons why most countries, not only developing countries, have not yet adopted this. But yeah, in the subsequent future, it will be available. Yeah, that's what I can say. As of now, it's in the pilot scale, it's in like studies, very few companies looking at not only biohydrogen, but also some intracellular pigments and molecules which are of like high value. And yeah, so, I mean, I'll just leave it at that. I'm not really that versed into like the political, you know, like why. I'm more into like the science and I can discuss the science. But the politics about that, I don't really know. Thank you.
No, thank you so much for your response to that. If any attendees would like to share their thoughts on this question, please feel free to share your thoughts in the chat box. Our next question is, in your opinion, to what extent can bioenergy replace fossil fuels in achieving climate goals?
To what extent?
Yes, to what extent can bioenergy replace fossil fuels to achieve climate goals?
That's a very good question, because when I think of extent, yeah, it's a good question. I cannot really pinpoint a number. It's something hard to predict, but I would say depending on the availability of the resources, because what is happening is the fossil fuel that we see today is actually what we are trying to do as the bioenergy, but it's just like it has accumulated for hundreds of years, you know, like thousands of years. So we are fast tracking that process and be able to like produce it as of now. So if we have the waste and there's resources in abundance, then we can almost cut off every, you know, like fossil fuel based source. But that is not an arena where people want to talk about now, because there's a lot invested already in the technology, you know, like in the resources, in the refineries, in, you know, like the crude oil industry. So right now people are looking at like, you know, hybrid kind of fuel, you know, like blending, you know, like using some bio-based and some fossil fuel-based, so we can slow down, you know, like the impact on the environment, and subsequently in the future, like replace it completely, because the resources are running out. So whether we like it or not, the fossil fuel will disappear. It will get finished. So in order to prepare now for the alternative form, we are now looking at this. But for me to tell you like what extent it can replace it, like pinpoint a value, it's hard for me to do that because I haven't done any study on that. I don't even know how that works, but I just know the fossil fuel resources are depleting, and we all know this. They're getting finished. So the oil wells are running dry in most places. They are trying to find new, but at some point in time, they'll be known. But we are also producing waste. What do we do with this waste that we are producing? That's where biohydrogen is coming in and bioenergy is coming in. So that's how I look at it from my perspective.
Wonderful. Thank you so much for your response to that question. We do have a few more minutes for the Q&A session, but if we do not receive any further questions, I will end the session soon. However, a last question from us to you is, since you have won the Research Impact Awards, what are your thoughts on what pointers would you like to share to current PhD scholars who would like to apply for this award in the future?
Okay. Yeah. So the first thing I would say is do a good work. Do a good work. Yeah, because when you have a good work, I mean, communicating it, it's not that difficult. People will see into it because I would say the judges actually go in depth into reading, into knowing like what you're doing. So if you don't have a good work, you don't really stand a chance because it's very competitive as per what I know. So what I would say is do a good work. If you're still starting your PhD, don't be in a haste to apply. Most people, first year of the PhD, it's mostly, you know, like literature review and intense kind of like, you know, grasping of skills that you need. I got the award when I was in my second year. So my first year work and like second year built up into what you see today. So this is not something that was just got in the first, you know, the first year of the PhD. So do a good work, take time in writing, you know, communicating it, and get validations from your supervisors because they're actually experts in the field. In my case, I actually consulted my supervisor, like I'm applying for this, and I'm like, this is my write-up, what do you think? And then my supervisor actually gave in some inputs. I think you should discuss more on this. I think this is too technical, keep this out, talk about this. So that helps already in the process of like, you know, conveying the message out because it's easy for us to understand what we are doing, but like putting it out there, it's also challenging. So yeah, have a chat too with your peers and get peer review, you know, like from peers, get review, get what do they say about it, you know. If they say good, it's fine. They say bad, don't be discouraged, take it and improve on it. Yeah, it's part of the process. I mean, that's what I can say. We get that all the time. I mean, among ourselves, we disagree, you know, like stuff, but, you know, no offense, no like, you know, like beef or like anything, but it's the work. It's like, it's supposed to be like this. This is proper science. This is thorough. Do it like this. And those feedbacks actually help you in improving your work, not just for the award, but the PhD itself, the whole experience. And yeah, so that's what I would say for that.
Wonderful. So constructive feedback from gaining and receiving constructive feedback is key to learning, I suppose, from your experience. Thank you so much for sharing your response to that question. And with this, we come to an end of our presentation.